So, firstly, the introduction part. Theobacillus ferroxidans, the bacteria that we choose. The species name is Theobacillus denificans. And Theobacillus ferroxidans is mostly live in an environment which has a high level of sulfur and ion, such as acid mine drainage, or sulfur-rich ores and mineral deposits. These bacteria have a specialized characteristic which can do autotrophic mechanism and chemolithotropic mechanism. So autotrophic mechanism is that bacteria obtain energy from for the growth and survival from carbon dioxide, but the chemolithotropic mechanism is the bacteria obtain their energy from the oxidations of inorganic non-carbon compound.
Nutrition type for this Theobacillus ferrocidans is autotroph. Theobacillus ferrocidans is an autotrophic bacterium that obtains carbon from carbon dioxide and energy from the oxygen in organic compound, such as ferrous iron and elemental sulfur. It is commonly found in environment rich in sulfur and iron, like acid, mine, drainage, and mineral deposit. Nutrition type based on carbon and energy source. Firstly, carbon source in organic compound with the example carbon hydroxide and bicarbonate. Second, energy source in organic compound which mean mineral and chemical reactions. Natural habitat, we have three sources, that is nitrogen, phosphate and potassium. Firstly, nitrogen. Autotrophic bacteria like Theobacillus ferrocidans can assimilate nitrogen from inorganic source like ammonia or nitrate present in the environment. Nitrogen compound can be found in the form of mineral or dissolved in water salts within their habitat. Second, phosphate, which means autotroph can derive phosphate as essential nutrients from inorganic sources like phosphate rock or mineral deposit present in the environment. These minerals serve as reservoir of phosphate that autotrophic bacteria can access. Lastly, potassium. Autotrophic bacteria can obtain potassium from inorganic sources like potassium mineral or the surrounding soil. These minerals gradually release potassium ions in the environment, providing a natural source of these essential nutrients from autotrophic organisms. In summary, autotrophic bacteria like Theobacillus ferrocidans derive carbon from inorganic sources and energy from the oxidation in organic compound. Oxygen requirement. Theobacillus ferrocidans is an example of an obligate aerobes, which means it requires oxygen for its metabolism and growth. It utilizes oxygen through aerobic respiration to obtain energy. To neutralize the toxic byproduct of oxygen, Theobacillus ferrocidans employs similar machine. Firstly, production of antioxidant enzyme, which means its product enzymes such as catalyze and superoxidize these motifs. Catalyze convert hydrogen peroxide, a toxic byproduct of oxygen metabolism, into water and oxygen. While superoxide, these motifs convert superoxide radicals, another toxic byproduct, into hydrogen peroxide and oxygen. Second, production of exopolysaccharide. Theobacillus ferrocidans secrete exopolysaccharide which form a protective biofilm around the cells. The biofilm acts a barrier against harmful reactivity oxygen species generated during oxygen metabolism. Lastly, repair system. The bacterium causes DNA repair machine that help in repairing of damage caused by employing these strategies. Thiobacillus spherocidans can mitigate the toxic effect of and thrive in aerobic environment, making it an important player in biobinding process where it contributed to the bioleaching of metal ores. pH requirement. Thiobacillus spherocidans has a specific pH requirement for its growth and activity. It is an acidophilic microorganism, meaning it thrives in acidic environment. The optimum pH range for Thiobacillus spherocidans is typically between pH 1.5 and pH 2.5. In this highly acidic condition, the microorganism can efficiently oxidize ferrous iron and sulfur compound. Temperature requirement. Thiobacillus spherocidans has a specific temperature requirement for its growth and activity. It is a mesophilic microorganism which may thrive in moderated temperature ranges. This optimum temperature for Thiobacillus spherocidans is typically around 30 to 40 degrees Celsius. Within this temperature range, the microorganism exhibits optimum growth and metabolic activity. Extreme in temperature, either higher or lower, can inhibit its growth and affect its overall performance.
Secondly, this bacteria are used in biomining industry. In biomining industry, Thiobacillus spherocidans is used to extract the valuable metals from the ores. Thiobacillus spherocidans is used in biomining for the ability to oxidize ferrous iron and sulfur compounds present in ores. It's utilized the process of chemolithotropic oxidation. It oxidizes the ferrous ion, which is Fe2+, and sulfur compounds to ferric ion and sulfuric acid, respectively. Now is the industrial application regarding this bacteria. Thiobacillus spherocidans is a bacteria used by most of the industrial for the production of their products or for many purposes. So firstly, we're going to discuss about Thiobacillus spherocidans is used by the brewing industry, which is specially for beer production. Thiobacillus spherocidans is utilized in in brewing industry for the role in sulfur reduction, which is this bacteria, specifically Thiobacillus spherocidans, is used to remove the sulfur compounds from malted barley during the malting process.
Conclusion. Thiobacillus spherocidans is a microorganism commonly utilized in the biomining industry for its ability to oxidize ferrous iron and sulfur compounds. It plays a crucial role in bioleaching, where it helps extract valuable metals from ores. Understanding the growth and metabolism of Thiobacillus spherocidans is vital biomining. By knowing its specific oxygen, pH and temperature requirement, industry can create optimal conditions for its growth, ensuring efficiency metal extraction. Additionally, understanding the metabolic process of Thiobacillus spherocidans, such as chemotropic oxidants, allow industry to leverage its capability to solidify metals from ores, making them accessible for extraction.